in Department of Materials and also chemistry. I've got two other roles. I'm also research area lead for the Royce in an area called chemical materials design. I'm also head of environmental sustainability for the School of Natural Sciences, and I'm also recently started as materials theme lead for sustainable futures. So I've got quite a few aspects, and it's really interesting, exciting for me to be able to connect in with different people. So yeah, I'm hopeful this meeting and this presentation will be of interest to people. I'm going to be talking about advancing long-acting therapeutics. I'm going to cover some materials design and some translational challenges. I'm also going to introduce the area. I think obviously this might be something that's new to lots of people, and also we've got a diverse audience who probably aren't really interested in nuanced material design aspects. So I want to try and give a flavour if possible. Okay, so let's think about something at a very high level background. So as anyone who's ever taken medicines, which is probably absolutely everyone on this call, the most common route of administration is oral, and that is because in part it's very convenient. It's used for a lot of treatment, so prophylactic, prevention, and medication of side effects. Typically this is daily, but in some times it can be several times a day, and often more than one tablet per dose. So it very much depends on the patient and what their kind of treatment and medications are. But actually in certain patient cohorts it can be over 10 tablets a day, and often as people get older they're more likely to have many drugs they're taking for a range of different conditions. So tablet numbers of over 15 and even 20 is sometimes possible. In terms of oral dosing, we can have tablets. There are liquid medications, and I've given some numbers here as well. We can just look at psychiatric medicines, typically at least 3, some heart disease and proton pumps, often 3 tablets, and then even trying to think about before meals, so when the person takes their tablets. Okay, so a bit of very basic pharmacology just to think about things. Here is your pharmacokinetics. So if you're not familiar with pharmacokinetics, it's a measure of the blood concentration, the drug concentration in the blood. So that's what your y-axis is here, and your x-axis is time. And so a patient's had a dose, they've taken a tablet, the drug's being absorbed, and as it's being absorbed, it's simultaneously being cleared. So you have a C max, a concentration at the max, and then it drops down. And you also have this therapeutic window, which is where we need to stay below upper threshold to avoid toxicity, and above a minimum threshold where we don't have efficacy. So this is our window we need to stay in. And then what we need is a patient to take another dose to keep it going. So here we can see a patient dosing up to steady state conditions, where they're in this window now, where, for example, each day they're taking a tablet and it's keeping them within this therapeutic window. Now, the problem is, the reality is that this hasn't happened for patients and people generally. So even 400 BC, we get this nice statement here: keep a watch on the faults of patients, which make them lie about taking things prescribed. Although not taking disagreeable drinks, purgative or other, they sometimes die. So essentially, as a doctor being prescribed things, patients aren't taking those. And we can flash forward to relatively recently, when the chair of the Royal College of GPs was saying essentially we prescribe people medications, we rely on the patients to take them, and they need to do that at a time and frequency that benefits them most, but they aren't doing it. So here's a little bit of data that kind of shows this. This is a real study on patients. So if we focus on the bottom part of the graph, this is time of day on the y-axis, and actually each day on the x-axis. So this group of patients was asked to take a tablet every day at 7 a.m. That's these orange dots. And the blue dots are actually when they took their tablets. So we can see that basically they never took one at the time they were supposed to take it. Sometimes they took 2. So here's 2 on one day, and the little kind of cream bars are when they took none. And then we go up to their drug concentration in the blood, and we can see the red line is the idealized steady state dosing, and the blue is actually what the patient would have had based on this dosing regime. So we could say here, because they took too many doses, they've gone into toxicity potentially, and here in this big break, they've dropped right out of the therapeutic window and taking time to get back up into it, which means that if they were taking a drug to treat, say, a virus or bacterial infection, you've actually got a sub-effective dose, so you can actually evolve resistance to your drug. So the reality is that people are very bad at taking their tablets. So we can see some examples: missed doses, multiple doses, very poor timing, and we can see the impact on the efficacy. When we think about the numbers are pretty striking in terms of what the effects are. Here for statins, adherence rates of 50% after 1 year, declining to 30% after 2 years. So 70% of patients are essentially not having effective treatment, and the cost of statin treatment across the UK is a sizable burden to the NHS, and most people are not really seeing the benefit. The one I personally find most shocking is this one here, which is around organ transplant. So 60% of organ transplant rejection and 30% of graft loss is due to poor adherence. And we can see some of the data for kidney and lung transplant. And then in the US, 22.5% of all organ transplants, failure is due to non-adherence. So even when people are taking things that are critical to their health, adherence is typically poor. The costs, and both in terms of lives and financial, are sizable. So within the EU, they estimate 200,000 avoidable deaths per year, and a cost of 125 billion, because of course anyone who is non-adherent is not having effective treatment from that drug, and therefore they have some of these ill adverse effects, which then further increase the cost of treatment. Okay, so just a few stats for the UK. Approximately half the population is taking at least one prescribed medication in the last 7 days. About a quarter taking 3 or more prescribed medications each day. And some people are taking, about 13% are taking painkillers every day. 30% of adults under 40 have taken prescription painkillers, and around half of the UK is taking prescription drugs. So we can see, if we imagine those numbers around adherence, clearly this is a really sizable effect if we think about how many people are taking drugs. An interesting one is around 14% of the population is taking someone else's medication. So where people are going, oh, this worked really well for me, give it a try. Obviously not really in line with the idea of, you know, taking drugs that are needed to give the benefit that are required. Okay, so the UK estimates are a bit lower. £500 million a year in avoidable healthcare costs is due to antibiotic poor adherence alone. So, I mean, yeah, this is again clearly a sizable effect. So let's think about what we could do. Now, obviously, there's lots of different ways you can try and improve adherence. There's apps on people's phones that remind them to take tablets. You can get these pill boxes that have the days in them, so people know which day they've taken and so on. But one route that's quite effective is a long-acting medicine. So here in the kind of grey line is our idealized daily dose of an oral tablet, and then in the blue line is our long-acting medicine. And so there's a couple of interesting things we spot. Is first of all, long-acting medicine should obviously get into this therapeutic window and remain in it for as long as possible. If we look at the shape of this curve drawn on as a cartoon here, we can see another thing that's quite interesting. So if we look under the line, every area in this pale red is more drug that's needed, because essentially in the blood, this drug is the difference between these two lines. But also, once we're within the therapeutic window, we don't need to be up near the top. If we can have a quite consistent release that's towards the bottom end of the therapeutic window, we can save all this drug in the light blue area, in the kind of peaks above this, above our long-acting dose. So, in terms of some of the benefits that exist for this, we can have this drug reduction up to 90% versus oral dose. And obviously this has a few benefits. It has a cost benefit. The API, the active pharmaceutical ingredient in any therapy, is pretty much the most expensive part. And that has a cost basis and also kind of a volume basis. In some regions of the world, there isn't enough drugs available, say for the treatment of HIV. It's hard to manufacture enough of the API, and so if you can use less of it to treat people, you can treat more people. We can obviously, if we can improve clinical outcomes, if we can stay in this window, we address those poor adherence issues I've talked about before. That also links into quality of life. And if you've got someone who's caring for someone else, it can have a big effect. So, for example, with psychiatric medicines, long-acting doses have improved adherence in psychiatric patients, which then reduces the burden for the family and carers around that person. We can extend the dosing windows, so it makes it more convenient for people. We can save on costs potentially, less drug, less packaging, and also fewer adverse effects can save on money for healthcare. And also another aspect is all around sustainability. I'll touch on this later on at the end of the talk. We have less packaging, and I'll give an example in a minute on the packaging example, but we don't have to have the blister packs, we don't have to have the cardboard packs associated with it, you don't have to have the shipping of all that packaging, and you don't have to have as much of the API, which also takes a lot of energy to produce. So there might be sustainability benefits as well there. So what do we really want in a long-acting medicine? We want something that's a single administration. So sometimes for some diseases, this could be a dose that's enough to give a cure. We want something that requires essentially no patient intervention. So the patient is given the dose and then they don't need to do anything else. The window it needs to replace depends on many considerations. It could depend on the patient needs, it could depend on the drug type, it could depend on what's possible, but you could generally, for our view, we think about replacing days all the way up to months. And we need the drug to remain circulating in the body for a long window as possible. So these aren't hypothetical. In terms of long-acting medicines already exist clinically, and I'll show another slide on some of the data in a minute. But we can see there's a whole range of different indication areas. So we've got antipsychotic, contraceptive systems, we've got hormone therapy, antibiotics, HIV, and another antipsychotic. So these are marketed by a range of different companies across the space. There's also, the US has a policy of administering all new recruits a long-acting formulation that helps with respiratory disease, so that's helped to reduce their numbers by 32%. And there's increasing approval now in the UK of HIV therapies. So NICE approved a long-acting HIV medicine in 2021, and then they also made a recommendation for prevention. So this was for therapy, and this was a similar formulation, but for prevention of spread of HIV. So for people who are at high risk, could take the long-acting medicine and essentially have or prevent being able to contract HIV. But one of the things is the actual scope of the clinical products is pretty narrow. So we can see that quite a lot is in the antipsychotic area. I'll talk about that in a minute. So what actually is a long-acting medicine? Well, there's a range of different types. So, and I'll be giving a bit more information on this throughout the talk. But one example is essentially we have an oil, or we have a particle formulation that we inject into the body, into either subcutaneous tissue or into the intramuscular region, and it forms a depot of the drug that slowly dissolves. Similar to that, we can have what's called an in situ forming implant, and I will definitely be talking about this because this is one of the areas we lead at Manchester, where we inject something, it's a liquid, and then when it goes into the body, it forms a solid implant that slowly releases drug over time. We can have microneedle patches. So here's an example of microneedle. You put it onto the skin. It disrupts the sternum corneum and allows drug to permeate through the skin, allowing drug delivery over time. More complex systems. So here's a refillable implant that's implanted in the arm. Drug is slowly released through a permeable membrane, and the drug can actually be filled over time, so you can keep the single implant in place. And there's also ones for post-surgical resection. So one area is in the brain, where post-tumor removal, you spray a gel, and that gel then slowly releases drug over time and essentially prevents the regrowth of cancerous cells at the site. So here's an example. So there's this formulation here. So this is work actually from the University of Liverpool. This combination of two drugs here that's being used for treatment. And in Liverpool, they managed to go. So actually, this is the way that this product is sold. It's sold as 3 tablets in one box, and it's, as it say, you see here, take through all 3 tablets once daily with food, at the same time with food. So you're taking quite large doses there. They've managed to concentrate and formulate that not as an oral tablet, but as a depot injection. And so for a patient to be treated, they need to have all these boxes. So this is a daily box. Each one of these are daily, and they need to take it for a number of weeks. So 6 weeks here. So we can see 6 sets of these boxes. And essentially in Liverpool, they've developed that formulation. So now that equivalent amount of drug is now concentrated into a suspension of nanoparticles that can be injected. So essentially replace all of this with one injection. So you can think about the convenience of not having to take 3 tablets a day, and you can think about also the saving in all the packaging and the transport of materials, but also for the patient, the lifestyle of if you live in an environment where you have limited ability to store these parts. So for people who have quite potentially disrupted lives, if you're given this big box of treatment, so this is for treatment of TB, by the way, if you're given this treatment, where do you store it? Where do you keep them all safe? How do you make sure you forget them? And you just replace that with a single injection. So this is obviously quite a sizable opportunity. Okay, so a few other bit of background information. So the first long-acting formulation, so LAI stands for long-acting injectable, was actually approved all the way back in 1952. And there's just under 80 that are approved clinically now. So this review breaks it down nicely. We can see that, first of all, if we look at the products on the market, it's been steadily growing. We can also see the different types of technologies. So there is quite a big diversity, but suspensions, which was the example I gave before, and oily solutions, then implants are the most dominant 3. And if we look at indications as well in this bottom graph, here we can see that schizophrenia being the primary one, and then oncology and palliative care second. But we do have quite a large diversity also in indications. So this is something that is quite diverse, both in terms of technology and disease area. So how do they work? They work by a process called flip-flop kinetics. I'll talk about an example of what that, well, I'll give you a simple analogy for what that is in a minute. We can save a lot of dose. So here's an example of some values on dose. So this is a HIV drug. Both of these are HIV drugs. And with cabotegravir, we have 30 milligrams a day. And so that means you take 1.7 grams of it over 2 months. A long-acting injectable is only 600 grams. So again, we're saving 2/3 of the dose. And then ripivirine, again, we take 1.4 grams over 8 weeks. And again, our long-acting injectable formulation is approximately half that dose. So here we can see more example of different dose reductions. Again, it depends on the API. So here's different APIs showing the reduction in daily dose that's possible from a long-acting formulation versus an oral formulation. So why can we reduce our dose? So oral bioavailability is one of the big ones. So when we take a tablet orally, it has to be essentially dissolved, and the drug molecules need to move across our gut into our blood. So that is primarily a barrier. So this absorption. We also get what's called first pass metabolism. So when a drug is absorbed through the gut, it goes straight through the hepatic portal vein into the liver, where it's metabolized. So you lose a big chunk of your API in that stage before it's even formally entered the circulatory system. And then ultimately, after that's happening, we've got the elimination. So how the drug is being either metabolized in other areas of the body or cleared through the kidneys is reducing our drug. So when we have a long-acting injectable or a long-acting formulation, we don't have these issues. So we don't have to go through the gut, so we don't lose this part. We don't have immediate first-pass metabolism because our drug can be administered, say if it's administered into the subcutaneous tissue, it enters the circulatory system from that site. It's not immediately metabolized in the liver. So actually our pharmacokinetics, PK, is controlled by the elimination rate traditionally. Whereas for long-acting formulations, they're actually controlled by the absorption rate. So a simple example of this is if you imagine a person is a bucket, and the bucket has holes in, so the holes represent your elimination and the water pouring in represents your absorption. For a lot of orally dosed aspects, you're pouring in a lot of drug, you've administered a lot, and then your pharmacokinetics is controlled by how quickly it's being cleared. You have to pour in a lot because you're taking a tablet once a day or twice a day, so you've got to have all that dose in that one window. So then the drug's already in the blood and gradually being cleared by elimination. Whereas if we imagine for a depot, we've got the drug in the body, but actually it's releasing very slowly. So we don't have to dump all that dose in. The dose is there, but it's only absorbing at very slow rate. And yes, we still have our elimination, our holes in our bucket, but actually the amount that's entering the blood is controlled by the amount that's coming in, so our absorption. So this is why it's called flip-flop kinetics, because normal pharmacokinetics is controlled by the elimination predominantly, whereas actually for long-acting, it's controlled predominantly by the absorption rate. Yeah, you can't eliminate drug until it is absorbed, is essentially the message there. Okay, so another thing is the relative interest from a community and policy aspect for long-acting interventions. So there's a few different studies out here. I'm not going to go into detail on these because this isn't my work. A lot of this has been led out of Liverpool, but if we look at some of the aspects, most patients are actually quite favourable. So this is for hepatitis C. Patients are quite preferring injectable systems if they offer benefits for them. In terms of policymakers, there is quite a high appetite for patient-preferred methods, and injection again being one of those. And an interesting thing is also these uptake of different technologies does differ a lot between countries, but here we can see an example of patient preference across different low and middle income countries, and we can see that in some regions the interest in patients of taking injectables in orange, or injections or patches, is really quite high. So in Ethiopia, India and so on, whereas in Egypt actually there's much lower uptake. So for us a big thing is also about understanding which drugs are most relevant for which markets and which indications. So, all right, let's get into a little bit more detail about what we're doing in this space. So back in 2024, 13.1 million was allocated from the EPSRC. So EPSRC was funding healthcare hubs. And so we were fortunate enough to be allocated funds to establish the UK's hub for Advanced Long Acting Therapeutics. So HALO is the abbreviation. So this is hosted at primarily the main hub of that is the University of Liverpool. And University of Liverpool has its own centre of excellence in long-acting therapeutics, CELT Global Health. So it's hosted there. And also we have Nottingham, ourselves in Manchester, and Queen's University Belfast. And we believe this is the first national hub in long-acting therapeutics, potentially globally. So it's a EPSRC focus, so engineering physical sciences focused activity. So it's very much focused on the fundamental physical understandings of long-acting medicines. Things we're doing is looking at proof of concept for long-acting therapeutics and focusing more on conditions where there isn't a long-acting option. So as we saw before, there's quite a lot in the HIV space and the psychiatric space, but also there's many areas where this adherence benefit could be really valuable. We're interested in extending the scope of long-acting therapeutics. And as part of our hub, we've set up a patient public involvement and engagement program. So this is in the UK to really understand the UK patients and the public's appetite for this, because there's no point us as scientists trying to push a technology or an approach that actually people aren't interested in. So we've got a survey launching. We might have actually launched in the last few days, but if not, launching very soon on that. We're engaging closely with clinicians and regulators, because of course there's some challenges around what do clinicians think about these systems and how are they regulated. And also we've been very active in trying to think about how we can support the ecosystem for translation. I'll talk about that in a little bit more detail, but obviously to make a new type of formulation, you need the right contract research or development organization to actually produce those formulations for further testing. Okay, so Halo has, as I said, 4 different universities. At the minute, it's across this kind of region between that spans across the Northwest. We have a whole range of different technologies I'm going to talk about on the next slide, but I think something we're really keen on is to expand the engagement. So we're really actively looking about how to engage with other organizations, both academic and industrial, in terms of empowering and spreading the opportunity for long-acting medicines. So Halo is disease agnostic, but it's focused on technologies. We have a range of technologies. So we have at Liverpool, we have some of the depot injections. So where we have nanoparticles that can be injected as a concentrated dispersion. At Nottingham, we are looking at gels for localized delivery for post-brain resection. So glioblastoma being one of the primary indications there. In Queen's Belfast, they've got an extensive background in microneedle patches. So these are essentially these tiny needles that just break through the top layer of the skin. They don't penetrate deep enough to trigger pain response. So they're essentially painless to administer, but by breaking through the top layer of the skin, the drug can diffuse through. And they've been trialing a range of different technologies in this space. And in Manchester, we're leading on in situ forming implants. So we inject a liquid, it forms an implant, and that implant provides long-acting release of a drug. So I'll talk about that in a bit more detail. So in Manchester, we have quite a diverse team. So we have myself and Alex, who are in the Faculty of Science and Engineering, and we also have Christoph, Cyril, and Marilina, who are across the Faculty of Biology, Health and Medicine. And we have a team of researchers who span different areas. So we have polymer chemistry, formulation design, cell work for in vitro assays, pharmacy and in vitro development. And Nora is looking at some life cycle assessment, which I'll talk about in some more detail later. So we have a really diverse team, and I think something we're trying to think about is how to connect those people more effectively. Our portfolio in Manchester is quite broad, so we have over £1 million in funding. So we've got 4 different strands of activity. So I'll talk about the in situ forming implant development shortly. We're also looking at in vitro models, so we can actually have systems that allow us to ideally rank different formulations to assess their translational potential. We're looking at some emulsions, so injectable systems as well that are long acting. And also we're looking at sustainability analysis. I'll touch on that later. So understanding what is the environmental impact of formulations. So what is an in situ forming implant? So here's a video, so we can see a liquid's being injected, and as the liquid enters, this is at body temperature and mimics the physiological environment, and we can see we form a solid implant, and that implant can contain the drug that then releases over time. So conceptually, an in situ forming implant is pretty simple. We have our API, I'm going to represent it very simply as a blue dot, and we have a polymer, which is my squiggly line here, and we mix those two together, and depending on our design, once we inject them, they will form some kind of solid, and that solid, or it could be a gel actually as well, then controls the rate of release, and our polymer can provide us the opportunity to have greater control than if we just injected the drug alone. So the current clinically approved systems of in situ forming implants are typically based on a process called solvent exchange. I'm not going to go into huge details on the materials aspect because I know we have quite a diverse audience, but just to kind of show some of the things we're interested in. So here's an example of the solvent we use, this N-methyl-2-pyrrolidone. So this solvent is water miscible. This is the solvent here, and allows us to dissolve our polymer. So this is our polymer that's used. It's called PLGA, and we dissolve a drug. This is ripivirine in this cartoon here. They're injected, and then we form our in situ forming implant, this lump here, and that lump slowly releases drug. We can see here, this is actually a cross section of this material, so it has some porosity in it, and the structure of the porosity and the relationship or interaction between the drug and the polymer influences the release behavior. However, there are challenges. So we get burst release. So the minute you inject it, some of the drug won't solubilize, won't remain, solubilized in the mixture and will come out, and that will lead to much higher release. And then also we have issues potentially around the toxicity of these solvents. So this N-methyl-2-pyrrolidone or another solvent that's used, DMSO, do have some concerns around their safety in vivo. So there are a number of clinically approved systems for this. They can provide release between 1 and 6 months. So we've got this formulation here that's in 2002, shown up here, which is for the treatment of prostate cancer. We've got other ones for opiate dependency, schizophrenia, and then also precocious puberty as well. So we can see that these technologies are already being marketed successfully and being used to treat patients effectively, but again, in a relatively small number of indications. Now, there's a number of different factors from the material science side that are really interesting. So some of the ones that have been reported before, people are very interested in controlling the rate of drug release, because of course, if we want to stay in that therapeutic window, we need to be able to release drug at the right rate. So we can do that through incorporation of additives that can lead to faster release, or we could vary the polymer properties. So there's a whole range of different kind of handles you've got that you can pull on these systems. Now, within our work, I'm not going to talk about the specifics because I thought it was too granular detail, but what we're looking at is a few central hypotheses that we're testing. So we're looking at our polymer. Can we control the microstructure of the polymer? So in here, what's the actual finer structure of the polymer? Can we use that to provide a barrier to diffusion or influence the diffusion of the drug through the polymer? This phase separation, so where the sample goes from liquid to solid, controls the burst release. So we're looking at the design handles, the design levers we can pull. So hydrophilicity is a big one in terms of controlling how that happens. And we also want to work outside some of the conventional polymers. So all the systems before are made out of this polymer, polylactic co-glycolic acid, PLGA, and we want to broaden outside of that to see what the effects of different mechanical properties and different plasticization behaviors are. Some of the other areas we're also looking at in our Manchester activity, a big one that might be of interest is in vitro release models. So currently, there aren't really good models to predict in vivo release for any long-acting therapeutics. We have poor representation of subcutaneous solubilization, mass transport, depot formation. This is all a very big gap. and that leads to weak IVIC, so that stands for in vitro in vivo correlation. Ultimately, we want to have tests we can do that allow us to quickly assess how a formulation might behave. We want to look at other systems. So one area is in the emulsion space. So can we look at new systems? So with new levers from a physical science perspective to control the way the drug is released. And a big one is also sustainability. So when we're making this comparison of tablets to an injection, actually we can only make an argument that one's better than the other with the data there. So there's actually a massive gap in data in this space. There is no comparisons reported of these oral and daily versus a long acting. So we're carrying that out. And then we also want to be able to think about what's the differences between different technologies, say an injectable that's a nano suspension versus, say, an in situ forming implant in terms of its environmental footprint. So I'm going to skip through. These two because I've talked about those and I've just seen the time. I'm just going to quickly talk about some of the challenges that exist for long-acting therapeutics from a formulation and technology perspective. I'm really thinking about the development cycle from formulation all the way up to commercial. So if we think about the formulation aspects, so some of the big gaps that we really have is that long-acting technologies are formulated in a highly empirical way at the minute. There aren't strong design rules other than knowing we've got to have the right kind of administration route, we've got to have the right indication, we've got to think about our drug and excipient design. But for example, understanding the relationship between the drug and the excipient is a big gap, and there isn't really predictive design frameworks in existence at all. That means there's a strong dependency on the different properties. So the properties of the drug, the excipient, the processing, all influence it. And then there's limited understanding of things about once we've formed a formulation, what is the structure of the formulation and how does that control the release? So again, these are big knowledge gaps that currently provide a barrier to developing better formulations. This is a big thing we're looking at in the project to try and drag out some of those design rules and really think about, how can we be much more predictive? So one of the things I was thinking about for us and thinking about the interaction with Pankhurst. So as you were saying before, there's lots of people in Pankhurst very thinking about AI and data. And I was imagining there's a whole range of different areas. So maybe digital twins for LAT systems. So can we model the systems and gradually refine the models to think about what is actually a good prediction that helps us understand the design rules as well. A big area we're thinking about, but having expertise on would be really useful, is how do we have our data infrastructure across our hub of all our different universities? How do we make sure we capture the data to allow us to compare things in a rigorous way and have systems that can be operable across the partners? Obviously, as we build this, we want to be able to think about how can we actually begin to do machine learning to predict the release. I think that's a big area. And another one is we've got aspects around microscopy, so where we're going to image our implants, and we have drug release studies. How can we actually fuse that data together? So fundamentally, we have experimental data, but we are missing this kind of connectivity. We're trying to work on this, but people with a stronger expertise in it would be a really useful link for us in terms of integrating our data. So let's think about our preclinical challenge. Preclinical challenge, in vitro in vivo correlation, IVIC is a big one. I've already talked around that about a lack of prediction between an in vitro and an in vivo experiment. Here's a recent study. So here they put long-acting microparticles in. This is their in vivo absorption on the y-axis. On the x-axis, their in vitro release. You can see that for some formulations, they've got a very good fit, but for other ones, there's big gaps there. This means it's quite hard to have consistent selection and testing in this preclinical stage to decide what you're going to move forward with without having to do every formulation in an animal. In terms of scale up, I'll touch on this very briefly, but obviously as you make a formulation in the lab, you have to be able to make that on a much larger scale to make it commercially viable. And any formulation any formulation is going to be sensitive to its processing conditions, and those processing conditions are potentially more complex or less understood for long-acting therapeutics. So here's a report from the FDA. They show some different formulations here, or repeats of formulations, and this is drug release over time, and we can see some variance just within that due to the differences that have happened between formulations. A big gap in the UK is there isn't much contract development or manufacturing organisations that have the capability to do the scale up. Regulatory pathway is challenging in some regards too, because there are gaps around, because there aren't that many long-acting therapeutics. There's some uncertainty about what data you need to provide. Again, think about your in vitro and vivo correlation expectations and comparing different formulations. There are also some routes to move through quite quickly if you can show you've got similar behaviours to existing formulations, but it is a challenging space. And then finally, from the commercial perspective, you've got high upfront development costs. You've also, so those are to do with the fact that it takes, if you've got a 3-month release, you have to do 3-month-long studies. All your studies are extended in their duration. So if you're going to have to do a clinical trial, you have to run those much longer than you would have to for an oral dose. You've got uncertainty about the reimbursement models, market size, patient acceptance. So we're trying to look at those currently through HALO. We need to try and get more clinician buy-in. So lots of clinicians aren't aware of the opportunity for long-acting medicines because they're not familiar with that opportunity. And we do have quite a fragmented ecosystem. So this is my second to last slide. I was trying to think about what could be some of the connectivities with Pankhurst and what could exist here really. And I kind of touched on that a little bit before, but this is thinking about the breadth of the project from our materials and excipients aspects. We've got a lot of data we're capturing. So we're thinking about a whole range of different parameters for our formulation. As we're capturing that data, what we really want to do is be able to link through the testing pipeline. So we can then, as we've made a formulation, we can then begin to test it. We'll have processing parameters as well. How do we capture those? We're not going to go into human trials in our study, but actually we're going to go into animal trials. And so what we want to do is understand what is our pharmacokinetics in an animal, and how do we pull out that data and feed it in. And often then, can we relate it to the economics and the real world aspects? So for us, I think thinking about this centralized data is actually a really big opportunity. We also want to try and have some cross-cutting principles. So we're trying to have more standardization across our hub partners. So again, we're all trying to use the same testing methods if possible, and make sure we've got traceability throughout that data. But I think, again, people in Pankhurst, I think this is hopefully an area that could be interesting. And for us, it's always thinking about what connectivity to add value. So with that, I'm going to stop. I went 47 minutes, so a few minutes longer than I thought it would. But yeah, obviously, I haven't mentioned really all the different researchers who've worked on this, but I tried to mention a few of the names on the Manchester side. We've got, I think, over 40 people in the hub across the 4 partners. So it's been really exciting to be involved in it. If you want more information, it's hosted here. If you just search the Centre for Long-Acting Therapeutics, it should come up. And if you've got any questions, either get in touch with me or you can also contact our central one there. And with that, I will stop.
Thanks, Tom. So